Testing, testing. Can people hear me? We're going to start at 10 a.m. my time, but just starting the stream now so that I can be sure everything's working as it should be. Awesome. You guys can hear me. If you hear any weird noises in the background, that's because I'm broadcasting from my apartment. So you might hear upstairs neighbors or a cat, and we're just going to deal with that today. How's everybody doing today? You excited to learn about some cool millipedes? There's about a 30-second to a minute delay on this, so we'll just deal with that, but it shouldn't be a problem today. Looks like everything is set up to work out, so I'm glad that everything is working so far and you guys can hear me. Again, we'll start at 10 a.m. sharp, so still about 4 or 5 minutes. Very glad to see all these pro-millipede messages in the chat. All right, well, looks like it's 10 a.m., so I'll go ahead and get started. Thanks everyone for joining today. Looks like we've got about 100 people here, which is super cool. It's going to be kind of weird that I can't see any of your faces and interact with you in the physical space, but thanks for being flexible and attending my virtual defense. So this is part of my, it's a part of the fulfillment for my PhD here at Virginia Tech, where I've been studying millipedes. We've got the chat here on the right, and this is where you can type in your questions at the end of the talk, and I will answer them to the best of my abilities. So I'm not going to be paying attention to the chat during my actual defense because that would be very distracting. But my advisor, Paul Merrick, is going to be taking care of the chat and sort of making sure y'all don't get too rowdy. So I guess with that, we can begin. So the title of my defense is The Natural History, Taxonomy, and Phylogenetics of Appalachian Flat-Backed Millipedes, particularly those in the order Polydesmida. These two illustrations here show two of my focal taxa, Nenaria on the left, Pseudopolydesmus on the right, and you should definitely check out those artists for more of their work because they are both just amazing in what they do. So to begin, here's a quick overview of how this whole talk is structured. I'm going to give you a little bit of introduction into millipede biology for those of you who aren't particularly familiar with millipedes. Then I'll go through each of the chapters of my dissertation and take you through what I did for each of those and the results that I got. And then finally, I'm going to have some brief summary and conclusions of the work, kind of the take-home messages from each chapter. As I said before, I am broadcasting this from my apartment, so if you hear any weird noises from like neighbors or the cat, please just bear with me. But hopefully Chloe will stay quiet during this whole presentation. So you might be asking yourself, millipedes? Yes. So millipedes are in the class Diplopoda, which is part of the subphylum Myriapoda. Their closest relatives are things like centipedes and also these two other groups called symphylans, the garden centipedes, and the poropods. The easiest way to identify a millipede is that it has two pairs of legs on most of its rings, and they're vegetarians, so they feed on decaying leaves, dead wood, things like that. They are pretty darn common in forest ecosystems, but you can even find some desert-adapted species, and all species are detritivores, so they are feeding on pretty much all the things that fall from trees, so dead leaves during the fall, things like that, dead wood, fungi, and they have this burrowing lifestyle where they're living down in the soil or sort of within dead wood. A lot of millipedes have really interesting chemical ecology where they use chemical compounds such as hydrogen cyanide, benzaldehyde, and quinones to protect themselves. Not all millipedes do this, but a good portion of them do. There are also many that are aposematic. They have this warning coloration to deter predation. Or you have the other end of the spectrum where they don't really have much color at all, which you can see on this photo of the right here, a species called Boetis carolinus. A really interesting aposematic trait that some of them have is bioluminescence. And this is something that my advisor, Paul Merrick, worked on as part of his PhD and postdoc. And it's a group of millipedes in California that can actually create light similar to fireflies, and that's used to deter nocturnal rodent predators. And so these are highly endemic detritivores. Many millipede species are not very widespread. They're sort of just local to certain places. Worldwide, there are about 12,000 species, and estimates of total species diversity goes all the way up to 80,000, but I think it's more likely that it'll be closer to 20 or 30,000 total species. And here in Appalachia, we're in a hotspot of millipede biodiversity, which makes it a great place to study millipedes, because a lot of this biodiversity is unique and unknown, and there are a lot of new species to go out and find and describe. So it's one of the best places in the world to study millipedes. And within that, the order that I'm focusing on today is the Polydesmida, the flat-backed millipedes, so called because they have this flat appearance to them, and you can see on the photo on the left and the right. The order contains 28 families with about 3,500 species, and they all have these paired and bicompartmental chemical glands, which I show in the middle here. And so these chemical glands are used to secrete a number of chemical compounds, including hydrogen cyanide, which acts as a potent poison to deter predation. And that's what I'm going to focus on today. My first chapter is about the evolution of cyanide defense glands and a Mullerian mimicry ring of millipedes. And so to start off, I'm looking at the family Zystodesmidae. These are commonly called the cherry millipedes. And here's an array of what their color is. You can see some of these species are brightly colored with yellows and reds. There are also oranges and other colors. And so pretty bright and colorful and just really nice and pretty when it comes down to it. But you can see B and F there are a little bit more demure in their color. They're a little bit more camouflaged. They're not really as striking. But what all these Zystodesmids share in common is this chemical defense. On the left here, I have a photo of their chemical gland after it's been dissected out of the body. It's composed of two parts, this large precursor reservoir that sort of looks like a large gossamer balloon, and then this thickened reaction chamber. And that is where an enzyme catalyzes a reaction that dissociates mandellonitrile into components including benzaldehyde and hydrogen cyanide. Benzaldehyde gives it this cherry or almond smell, hence the name cherry millipedes, while hydrogen cyanide acts as a potent toxin to predators. And you can see in this image on the right where the gland is within the body of the millipedes. It's right on the edge of what we call the perinoda, those horizontal extensions from the body, and about half of the rings in the millipede's body have these glands. Now, we're going to be talking about mimicry here, and you may be familiar with the two main types. Mullerian mimicry, where there is a shared signal of some sort, whether it's color or sound, as well as a defense, and these are both honest signals. In a Mullerian mimicry ring, two or more species will evolve towards a maximized signal, so similar color patterns, such as in mimetic advergence, and that's based on the genetic variation that is just within the DNA of the millipedes that sort of allows for natural selection to act on this. There's also Batesian mimicry, where there's this shared signal, but some of the species are non-toxic, so that's a dishonest signal. And so I know that growing up, I learned that the differences between these were presented as sort of a binary, but when it comes down to it in nature, it's more of a continuum between the two. And so that's visualized in this graphic here on the left, where you can think of the red being defended species, whereas white is undefended. And so there's a gradation between the two. And so this has been elaborated on by different studies. And so we also have states between the two that are called quasi-Batesian mimicry, quasi-Mullerian mimicry, and super-Mullerian mimicry. And so these are sort of different states between those, where it's not so clear-cut as one or the other. And so you might be asking yourself, well, what are some of the millipede predators? We don't have a ton of great data on this, but we do think that some of the vertebrate predators include birds. You can think of something like a turkey, where they're scratching at the leaf litter, and they're going to be likely to encounter millipedes, as well as things like toads in the genus Anazyrus. And there's also an unknown predator that has been dubbed Robespierre because it apparently decapitates millipedes, and then, millipede researchers will come across these beheaded millipedes, but we don't know what predator is actually doing that. And so when you think about it, the signal that the millipedes are using is likely going to be keyed into the visual systems of these predators. And so what then makes Dissertation millipedes a good study system for mimicry? A very large part of this is that Zistodesmidae lack eyes, and so they're not seeing each other, so we can be sure that these color patterns are not meant for sexual signaling, so they are geared just towards the predators. They're also abundant where they occur. In one example, there's been 43 individuals of 5 species found in an area no larger than 50 square meters, and so they're relatively easy to find in the field when you get good habitat. There's also no known diet-collar correlation. So these millipedes are synthesizing these collars in vivo. They're not just taking them from their food. And we know of a mimicry ring in the Cumberland Plateau area of Southwest Virginia and adjacent Kentucky and Tennessee, based on some work that Paul did during his PhD, where he described this Mullerian mimicry ring between Aphalloria polychroma on the top here, called Aphalloria clade because it wasn't described at the time of this publication, compared with sympatric species of Brachoria, which share collar patterns. And so he found that multiple collar morphs and patterns are found within species, and that these patterns vary geographically depending on which species co-occur, which is really interesting and just beautiful when you get out in the field and you can see these things. And the 4 main conclusions from this paper were that these millipedes shared an aposematic signal, which cannot be attributed to close evolutionary relatedness, and that rarer species tend to mimic the most abundant or most highly defended model. In his work, he found that to be Aphalloria polychroma, which was acting as the model, and that this mimicry varies geographically as a function of the model's geographical variation in collar pattern. So essentially, where Aphalloria is, the sympatric Brachoria species that mimic it will share those collar patterns, but then in other areas of the Brachoria range, those collars are different. And so we know that the model Aphalloria is having an effect on the Brachoria collar patterns. And so to summarize this, the aposematic patterns vary sometimes quite drastically in the species' geographical distribution throughout Appalachia, particularly in this Cumberland Plateau region. And these patterns are similar locally and have this mimetic resemblance. And that this collar mimicry also occurs between this widespread model, Aphalloria, and these narrowly endemic mimic Brachoria. And, this is interesting. And this parallels more common Mullerian mimicry rings that you may have heard of, such as heliconius butterflies in the tropics and poison dart frogs. So, within all that, what I wanted to do with this project was to examine the cyanide gland size within this context of Mullerian mimicry. And so my hypothesis for this project is that Aphalloria, being the model for this Mullerian mimicry complex, will have larger cyanide glands, and that Brachoria is the mimic and will have smaller glands. So what this translates to is that Aphalloria with these bigger glands are going to be more toxic, whereas I think that Brachoria will have smaller glands and thus be less toxic, so they don't have to invest as much energy into making all these toxic compounds, which are pretty intensive to make. So my objectives here is to test if the model Aphalorhea polychroma possesses these larger chemical glands based on the size of the reservoir chamber, where all of those precursor chemicals are stored. And within that, also test a prediction of dishonest signaling, that the model species will have these larger cyanide glands, and over evolutionary time, the toxicity of these mimicking Brachoria species will have declined. So, I've got 4 main methods as part of this, which include field collection, actually measuring the glands themselves, phylogenetic inference on a tree, and ancestral state reconstruction. And so this image here shows a pretty common forest where we would look for millipedes, and really looking for millipedes is great field work, so it was a lot of fun to get out and actually look for them. And another example of sort of common habitat that we see here in the Appalachians for these cystodesmids. A very nice thing about this project is that we had a large amount of material already in the Virginia Tech insect collection, so I was able to immediately get started on dissecting out these glands and measuring them before having to go and collect more specimens. When I did go into the field to collect, I targeted these moist deciduous forests, and I used a standardized procedure outlined by Means et al. 2015, essentially using a millipede rake or a garden claw to flip over leaves and logs and expose these millipedes. And then I brought these millipedes back to the lab to photograph them and document what their living color is like, because once you preserve them in the alcohol, they lose their color. Now, as for collection localities here, on the left is an inset map from Paul's revision of the genus Brachoria, and it shows all the species that occur in Southwest Virginia and adjacent areas. So you can immediately see there are a ton of Brachoria species in this area. On the right, I have the 6 sites that I use specimens from. And so these were all sites that had documented shared collar patterns between Aphalorhea and the sympatric Brachoria species. I also wanted to get a wider array of specimens to measure from across the phylogeny of the Zystodesmidae in the eastern United States, and so this map shows all of those collections. I collected an additional 12 species for this project to see what the sizes of the cyanide glands are like, kind of throughout the tree of Zystodesmidae. Now, as for the gland measurements, to even visualize the gland in the first place, you need to digest the 12th body ring and trypsin. And so the gland is derived from the exoskeleton, so you can dissolve all the muscle and everything around it with trypsin, and it just leaves the gland, which is pretty handy. I measured 7 to 30 specimens of each species, depending on how many specimens were available. Took a photograph, which you can see on the right. I show an example of an Aphalorhea polychroma gland and a Brachoria sedra gland. That scale is 2.2 millimeters in both of them, and so you can see already that this Aphalorhea gland is a good deal larger than the Brachoria gland. After I took the photo, I outlined the gland in Adobe Illustrator and then measured the area of this precursor chamber with image J. And then took a weighted average of gland size to use that for each species to plot it on the phylogeny. And these measurements were natural log transformed to account for body size variation. That way we can sort of standardize it. And then I looked in particular at Aphalorhea and the sympatric Brachoria species and compared them with a 2-sample t-test to see if there were significant differences. For the phylogeny estimation, I used a published phylogeny from Means and Merrick 2017 here on the right, and Brachoria is up here, so I chose a couple species from there. And this is a Bayesian phylogeny of 6 mitochondrial and nuclear genes. I then used this phylogeny to do a least squares parsimony ancestral state reconstruction, where I'm plotting or inferring the state of the size of the cyanide gland at each node within the phylogeny. And so I wanted to make sure that the species were independent from each other, so I selected species from several different genera and across the various clades within the eastern North American group of Zystodesmidae. After I did that, I then calculated phylogenetic distance by adding the branch lengths that from the root of the tree to each reconstructed species node. So this is the full tree. The collared branches are the specimens that I selected for analysis in this chapter. And so you can see 15 collared branches on this tree. And so that kind of shows the distribution of the species that I chose from the phylogeny. So for my results here, in total, I collected 15 millipede species from 11 different genera and 4 tribes within the Zistodesmidae. I measured a total of 222 individuals, including 3 species that are from documented mimicry rings and 12 species that do not have documented mimicry rings. It's important to note here that these ones that don't have documented mimicry rings, that doesn't mean they're not involved in a mimicry ring of some sort. It just hasn't been assessed. So these are the results of the reservoir chamber area between Aphalloria and 2 of its sympatric Brachoria. And so I've got up top here in these boxes the number of specimens that were measured for these, and then the white text in each of the bar just tells you exactly how large it is. And so what we can see here is that Aphalloria has cyanide glands that are double the size of either Brachoria species. And this is found to be significantly different from the other. So from this, we can conclude pretty well that yes, Aphalloria does have these larger cyanide glands, and it is much more toxic than the sympatric Brachoria species, which is pretty cool. Aphalloria was found to be the model before, but the exact toxicity of it hadn't been measured, so this was really interesting to see. And then to show the ancestral state reconstruction, this is the unpruned tree that includes all species, including the ones that I didn't sample. And then pruning this tree down to just the sampled species, we get this tree. And so the blue branches are the smallest branches, whereas the red ones are the largest cyanide gland size. You might be asking yourself, why are some of these values negative? Just remember that these are natural log transformed, and so that's why. Now, at the base of the eastern clade here, we have this blue bar for Giallostethus monticolens, and that's a pretty basal species. And you can see throughout the rest of the tree that the cyanide glands get larger, particularly here in the tribe Aphaloriinae. I put an arrow to where the Aphaloriinae starts, and you can see throughout these we have a couple red and orange branches there, whereas in the more basal parts of the tree, you sort of have these smaller glands. Now, plotting this out, these are all the 15 species that I measured. Again, you can see Giallostethus down here. And on the y-axis, we have the cyanide gland size, that area of the precursor chamber. And then on the x-axis, the root to tip distance. And so essentially, we're looking at escalation of the cyanide gland size throughout the phylogeny. And what I found was that the gland size is highly correlated to the phylogenetic distance. And so those Aphaloriinae are all clustered up here in the top right. And so we can see that over evolutionary time, the size of the cyanide gland has increased, in some cases quite dramatically. Now, directly comparing the Aphalloria to its sympatric Brachoria, what these graphs are showing are the chemical gland size, and then that root to tip distance on the x-axis again. And this is taken from their most recent common ancestor within the Aphaloriinae. So we wanted to look at where these species diverged and what happened to their gland size. And so we can see all of these graphs are highly correlated. In Aphalloria, we have a quite large escalation in the gland size, whereas in both these species of Brachoria, the gland size decreased, again, sometimes dramatically. And so we can see that, again, Aphalloria is more toxic, whereas Brachoria, these two sympatric species at least, became less toxic. Now, we also did a little bit of work with the scanning electron microscope. I tried to look at the reservoir chamber, but unfortunately, since it's so just kind of thin and gossamer like a balloon, you can't really see much on it under the SEM. But this is the reaction chamber. And what we see here are these two types of pores within the chamber itself. This hole type pore, and then the salt shaker type pore. And so the salt shaker pores, they can have anywhere from 2 to 8 holes kind of set in these mounds like a salt shaker. And this, I assume, is where the enzymes are released into the reaction chamber. And so that was really interesting to see. I'm not aware of any other SEM work with these cyanide glands, and so this was just really interesting, and it might be a great area to look into in the future to research the toxicity of these millipedes. So, looking back at my objectives, I wanted to test if the model Aphalloria polychroma has larger chemical glands. And we can say that yes, Aphalloria has larger glands and it's more toxic than its sympatric Brachoria. Secondly, I wanted to test a prediction of dishonest signaling, that the model species will have these larger cyanide glands, and over time the toxicity of the mimic declined. And I can say again, yes, Brachoria shows an asymmetric divergence in its cyanide gland size, fulfilling this prediction. So for some conclusions for this chapter, I've got here a list of the species with the largest gland size, Brachoria initialis, Sigmoria nantaheli, Aphalloria polychroma, and 2 species of Dixioria. Now, aside from the Aphalloria polychroma, none of these species co-occur with any Aphalloria species. So it's possible that they lack a protection conferred by mimicry ring, and so maybe that's a reason why they have such large gland size. And so this would be really interesting to investigate throughout the entire phylogeny of these eastern zystodesmids to see if mimicry rings influence the cyanide gland size as a general rule, or if this relationship we're seeing in the Cumberland Plateau species is unique. So, to summarize, Aphalloria polychroma, this model, is more toxic than the sympatric Brachoria species. These sympatric Brachoria species have evolved lower toxicity over time, and so they're acting more as quasi-Batesian mimics rather than full Mullerian mimics. And allopatric Aphallorians, those species that do not co-occur with Aphalloria, have larger cyanide glands. Now, I want to move on to my second chapter here, where I look at the genus Ninnaria, particularly this Ninnaria wilsoni species group. This group of millipedes is called the twisted claw millipedes. This center photo here is a micrograph from an SEM showing the twisted claws of the males from where they get their names. So this genus was described in 1918, and it's a group of cryptic and rare millipedes. These are also in the family Zystodesmidae, but they're not flashy and showy. Their collars are a little bit more subdued, and they're a little bit more soil dwelling. They're also not as abundant in the forest, or at least they're more difficult to find. Typically, when we go out in the field, we can pretty easily find the larger Zystodesmids, but these guys, we might be lucky if we can find about a handful or so. So why would you want to study Ninnaria? Well, it's a really interesting case study in Appalachian endemism and speciation, which is something that the Merrick Lab here at Virginia Tech studies. They are widespread in eastern North America. I'll show you a distribution map later, but they occur all the way from Arkansas to Mississippi, north to New York. And there are 23 described species, but there are many more that are undescribed within museum collections. So these illustrations here are from Richard Hoffman, who was previously at the Virginia Museum of Natural History, and he did a lot of illustrations of the various gonopods of Ninnaria. Gonopods are these modified legs that the millipedes use for mating. And so just from these illustrations, we knew, oh, there are a lot more undescribed species that we don't know about. Now, what made this a nice project to work on is that I worked with my colleague Jackson Means, who finished his PhD in 2018 on this. And so this is a phylogeny from his dissertation where he found that there were 2 groups within Ninnaria, a wilsonite group and a minor group. And so the minor group here is in green, and Jackson found 36 new species within that group, which is just kind of mind-blowing. The Wilsoni group is up top here, this sort of purple highlight, and we weren't sure how many new species were in this group, but there are only 6 of them that have been described. And so this was a great area to tackle, and the field work for this involved me and Jackson going and collecting a ton. So my objectives here were to investigate the evolutionary relationships within an area and describe these new species that we knew about. I also wanted to test if Ninnaria is monophyletic, and also estimate its evolutionary history based on 6 genes and compare that to the morphology and see if these species concepts line up. So I had 3 main parts to my methods here, the first being fieldwork, then working with museum specimens, and a whole lot of molecular phylogenetics. This photo on the right here was taken at a nature preserve in Ohio. You can see Jackson there with his red backpack, which made him very easy to find in the woods, so I always appreciated that. So again, with this fieldwork, I targeted moist deciduous forests. Here on the right, this is a rhododendron cove from Jackson County, North Carolina, and these were the types of areas that I would focus on, since these species are found in pretty mesic areas, so you want to be able to find moist leaf litter and microhabitats for them. For the molecular phylogenetics, I would remove the legs from the millipede to use those in DNA extraction and sequencing, and store those in the -80°C freezer in the lab. I extracted DNA using the QIAGEN DNase kit and targeted 6 genes. 2 of them were mitochondrial, CO1 and 16S, and the nuclear genes EF1 alpha, 28S, FBOX, and RNA polymerase II. I then inferred the phylogeny using maximum likelihood methods and Bayesian inference, and aligned the sequences with the program Mesquite, and then analyzing it in the program Mr. Bayes. So I want to talk about the museum collections that I worked with for a little bit here. We had great collaboration with the Virginia Museum of Natural History and the North Carolina State Museum, who both loaned us all of their holdings of Ninnaria, which totaled a little over 2,000 specimens. So it was a real treasure trove. I went through and digitized and georeferenced all these specimens and put labels within them, and use that for distribution maps and figuring out where to collect. So as far as a distribution for the entire genus Ninnaria goes, I've shown that in this white highlight here. And so the black dots are the species within the Ninnaria wilsoni group. So wherever you don't see black dots, you can just assume that that's where the Ninnaria minor group is, because they occur throughout the entire range. You can see the furthest west they get is Missouri and Arkansas, and they get north almost to Canada. They might occur in southern Canada, but we just don't have any records for that. Now, what's interesting here with the Ninnaria wilsoni group is that you can see there's this distinct break in the distribution. And when I was first investigating this group, I thought, oh, maybe that's just like a collecting artifact or something. But after repeated attempts to go to this intervening area and find any Ninnaria wilsoni, it's not an artifact at all. That's the actual distribution. There are no Ninnaria wilsoni in this area of northeast Tennessee and northwest North Carolina, but there are multiple species of Ninnaria minor there. So we have this interesting break in the distribution for some reason. You can also see that Ninnaria wilsoni is also restricted just to Appalachia. The furthest west specimens are here at Powell County, Kentucky, and here in Tennessee, but also along these mountain ranges, so part of the Appalachians. Now, to zoom in on this northern distribution here in Virginia, West Virginia, and Kentucky, I have the dots here are the distributions of the described species, and then the stars are undescribed species. And so you can immediately see there are a ton of undescribed species, particularly here in Virginia. Only 4 of these species are described, and there are 11 more to be described here. So this was really interesting to see. If you're not as familiar with Virginia's physiography, kind of this distribution up here is along a mountain range, and then there's another mountain range here, whereas the Central Valley is kind of between that. You can also see some interesting vicariance here by the new river, and I'll highlight that a little bit later. Looking at the distribution of this southern group, again, we see that same pattern where only 2 of these species have been described, and we have 7 undescribed species. Northern Georgia, in particular, is just full of undescribed species, and I'm positive that there are more there that we just weren't able to collect. So definitely more collecting in northern Georgia, and likely adjacent Tennessee as well, will likely turn up more new species, which is super exciting. And so the results from my field work, I was able to collect 374 specimens in 7 states. So I was able to combine these with the museum material that we already had to really check out the entire distribution of the group. I would find these millipedes, particularly, they would be soil dwelling rather than within the layers of the leaf litter. They kind of have their backs exposed at the top of the dirt. And especially in Tsuga and rhododendron coves. So rhododendron, Tsuga is hemlock, but they could also be found in oak, maple, beech, and tulip tree forests as well. Jackson and I, after a couple years of collecting and sometimes not having luck, sometimes having more luck, we eventually keyed in on that we need to be checking in these moist microhabitats. So this is an area in northern Georgia. You can see there's a stream down here. And if you look close, you might be able to see in this spot and this spot here in the center, I've moved a lot of dirt around. And so that's where I ended up finding Ninnaria millipedes there. So they like these pretty moist habitats there. Now to move on to the molecular phylogenetics. This tree here, there's a lot going on, but I'll zoom in on the important parts in a bit. But for now, just know that this is a Raxamel maximum likelihood tree of 1,000 bootstraps with those 6 genes I mentioned earlier. And one of the main takeaways here is that in orange, we have the Ninnaria minor group, and blue is the Ninnaria wilsoni group. And so I'm also finding support for 2 bit large clades within the genus Ninnaria. And so this is expanding on some of the previous findings that Jackson Means found. Up here are the outgroups. Green, which is a little bit tough to see, is Enamia pulchella, the sister species to Ninnaria. And then purple is Uri merodesmus, and red is the Uri urina, the most closely related groups. Now, looking just at the Ninnaria wilsoni clade, I have colored the branches according to the species here. And so this is that same tree, and I have all 6 described species within the group on this tree, as well as 18 new species. The asterisks here indicate a well-supported branch, that is bootstrap support of over 70%. And you can see that the species relationships are pretty well supported for the most part, but these sort of higher relationships of how the species are related to each other are not as well supported. I currently have a Bayesian tree running, so I'm hoping to get a little bit more insight from that tree and compare it with this Raxamel tree. Now, we found those 2 clades phylogenetically, but I also wanted to investigate if anything about the morphology of these groups stuck out. So again, we're looking at the gonopods here, those modified legs that millipedes use for mating. On the left, I have the wilsoni group, and on the right, I have an example gonopod of the minor group. And so some of the main things that I found between these groups are that this main branch, called the acropodite, often has these interesting twists and helices and sometimes other lobes and processes at the tip, like you can see here. The secondary branch, which we call the prefemoral process, originates at the base of the acropodite. In the minor group, we see a little bit of difference. The prefemoral process here is attached to this acropodite, and it originates from a shelf of the acropodite. There's also this elongation between the base, the coxa, and then the prefemoral process origin, which we do not see in the wilsoni group. And so the male gonopods are the main method for identifying and differentiating species of millipedes, particularly within the Polydesmida. But I also wanted to examine the female genitalia and see if there were differences there. And intriguingly, I was able to find a couple. And so what we have here is the second pair of legs removed from one of the Wilsoni groups. And you can see I've got a coxa labeled there, and these coxae are very much expanded, which cover the openings of the genitalia in the Wilsoni group. And we don't typically see that in the minor group. Additionally, the cyphopods, which I have down here below the leg image, have some really interesting modifications, particularly to the structure called the receptacle. And this particular species, it's expanded to this curving finger-like projection that just totally blew my mind when I first saw it. Typically, the receptacle in Dissertation millipedes is just sort of a quadrate flap, but this seems to be species-specific and seems to hold over different specimens. So that's definitely something that I've been looking more into. And so the main part of that is that we are seeing within an area both genetic differences between these clades and morphological differences as well. As far as color variation goes, this is pretty much all you see with Ninnaria. They're going to be this dark kind of chestnut brown or black with perinodal spots on the sides that range from orange to pink to red, or they can be white like we see in this top right photo. But that's pretty much it, and that's fine. It makes Ninnaria pretty distinct, and we never see these sort of mid-dorsal spots like we saw in some of these more colorful cherry millipedes. Now, the gonopod variation in the Wilsoni group is pretty interesting. Sometimes they're pretty simple, like this second one from Craig County, Virginia, but they can be pretty elaborate. This one from Raymond County, Georgia, almost has this sort of unicorn horn coming off of this main branch. They have this pretty distinct twist there. And so these were pretty interesting gonopods to look at. And so to really study these gonopods, what I would do is put them under a very nice camera system that we have in the Merrick lab and photograph the gonopods from 3 standardized views. For example, the view you're seeing here is the interior view. And then to describe these species, I went through and illustrated all of those views. So what that looks like is this. And so these are the illustrations of each gonopod that I use in diagnosing the species and also in comparing the species to each other. And so I made sure to use the left gonopod as a standard so we can really compare and be sure when there's differences between these gonopods rather than it just being like a difference of angle. And going back to some of the interesting ecology of these Ninnaria, this is a zoomed in view of Southwest Virginia. Blacksburg is up here near this cluster of red dots. And so on the east side of the New River, we have 2 species, Ninnaria ericacea in red and Ninnaria wilsoni in blue. These species are never found west of the New River. And so they really seem to be limited by the course of the New River there. Whereas once you get to the west side of the New River, there's this new species that we're dubbing Enigma. And so again, Enigma is never found on the east side of the New River. So this would be a really interesting phenomenon to study and kind of go all along the east and west side of the New River to see if this changes at all and if it might be reflective of changes in the course of the New River over time. So there are a lot of interesting biogeographical things that could be investigated with this group here in Appalachia. So to sort of bring everything together, I've confirmed this discovery of 2 distinct clades within the genus Ninnaria and identified 18 new species, which triples our known diversity for the group, which was just mind-blowing. So now we know of 24 species within this species group. And additionally, this group is restricted to Appalachia. You can see in this image on the lower right here, the sort of physiographic regions in the area, and they're really tightly restricted to just these sort of mountainous areas rather than these lower Piedmont areas. And so with all this investigation based on the genetics, based on the morphology, the biogeography, it seems likely that this Wilsoni group might need to be split into its own genus. So to summarize my objectives that I had here, I wanted to investigate the evolutionary relationships within an area and describe new species. I found 18 new species, and I am in the process of describing them. So I think that I can consider that a success. There's certainly more new species in the group to be found, but you need to stop sometime and actually publish it. So this was our stopping point. I also wanted to test to see if Nenaria is monophyletic, and it seems likely that we can split the genus into a separate one from the minor group. Additionally, I wanted to estimate the evolutionary history from 6 genes and compare the genetic results with the morphology. And overall, the morphology and the molecular phylogenetics are pretty congruent. And so that was pretty nice to see. So with that, we've reached chapter 3 here, where I will be talking about the taxonomy of Pseudopolydesmus, sometimes called the pink flat-backed millipedes. So again, this is the order Polydesmida, but I'm looking at a different family this time, the Polydesmidae. These are basically holarctic, and there are over 240 described species, but the taxonomy is in need of a lot of work. Within North America, it's a little bit simpler, particularly here in the east, where we only have 2 native genera present, Pseudopolydesmus here on the left and Cytinotus on the right. In Western North America, there are at least 6 additional genera, with more being described based on the work of Bill Shear and some collectors up in the Pacific Northwest. So that's really exciting, and it'll be interesting to see how many new species come out of there in the future. Now, we'll be focusing on Pseudopolydesmus here, and this is a pretty common millipede. When I was first getting interested in millipedes up in Ohio, this was probably the one that I was encountering most often. But it's kind of grown on me, even though it's pretty common. Here on the right, this is an image of a female, and they have this interesting behavior where they'll lay this sort of donut of eggs, which you can see here in the middle, and then they will build this dome out of feces and mud to surround the eggs and kind of protect them until those eggs hatch. Males also have these interesting structures on the legs called spherotrichomes. It sort of looks like a narwhal head with a thumbprint on it. And so those were really interesting to investigate under SEM. Now, why study Pseudopolydesmus? Again, it goes back to the goal of the Merrick Lab here at Virginia Tech. We want to investigate this Appalachian biodiversity. And these are some of the most common millipedes in eastern North America. But even though the first species was described about 200 years ago, we still don't have a great classification for the genus. And it wasn't until last year that we had a suitable morphological classification for the genus. So looking at Pseudopolydesmus, it's relatively easy to tell when you have a male, because if you flip it over, you see these normal legs, and then you hit this area that just looks very strange. And so these are the gonopods, and I have one of them dissected out here on the right. And this is the typical plan for Pseudopolydesmus, where it has just a single branch and it has a couple of triangular processes along each side of that main branch. So since the first species was described, there have been 39 names assigned to the genus, with most of those ultimately being synonyms. Last year, Paul and I collaborated with Petra Searwald and Xavier Zonley at the Field Museum of Natural History, and we narrowed this down to 8 species in the genus based on the morphology. There have also been 2 morphological groups proposed for this genus. One, the Canadensis group on the left here, which typically have larger and higher number of processes along the gonopod, and then the Serratus group, which is a little bit more of a simple gonopod. But these have not been tested phylogenetically. So my objectives here were to investigate the evolutionary relationships within Pseudopolydesmus and resolve these species boundaries, define the geographic distributions for each species, which have been pretty general in the past, test the hypothesis that Pseudopolydesmus is monophyletic. It was previously split into 2 genera, and so using genetic data, we can test that pretty robustly. and estimate its evolutionary history from 5 genes and compare that with morphology to see how well they match up. So again, this project took a lot of field work, working with museum specimens, and doing molecular phylogenetics. I followed the same molecular phylogenetics process as for Nenaria, but this time targeting just 5 genes, CO1 and 16S, and then EF1 alpha, 28S, and enolase. I also estimated a species tree using a multi-species coalescent model and StarBeast. Now, we also wanted to do a little bit more with integrative taxonomy with Pseudopolydesmus. And so there's a great paper published in 2014 by Jakara Betian and Hennen, where they used a series of steps to describe new species of harvestmen out in Western North America. And so essentially they were using the morphology to come up with species hypotheses, and then testing that with genetic data using StarBeast and then validating it. So this was sort of the game plan I followed for Pseudopolydesmus. So within that, I measured 15 different morphological characters in 69 male specimens of Pseudopolydesmus, and I accomplished that with the help of an undergrad, Maddie Hellier, who did a lot of great work with that. I then used these measurements to run a modified principal components analysis, and then did this analysis in R. So the results of this field work, I collected almost 1,200 specimens in 20 different states here in the eastern US, and targeting mixed deciduous forests, kind of like I did with Nenaria, but also targeting some floodplain forests and even swamps, because there are a couple species in the genus that are swamp adapted. If you look closely on the left, all the green leaves that you see on the ground are stinging nettle, and as any field biologist knows, stinging nettle always hides the best stuff. So that was the case here. I found a species I've been looking for a while and only had a little bit of stinging pain in my legs from the nettle. To show you a map of exactly where I collected, we visited 300 different localities, and again, almost 1,200 specimens. So there was a ton of field work involved with this project. Now, again, I worked with the Virginia Museum of Natural History and the North Carolina State Museum. They lent their entire holdings for this genus, and that amounted to almost 5,000 individual specimens. So it was a ton of material to work with and a great help. I also went back through the literature and digitized and georeferenced almost 1,100 records that encompassed about 3,300 specimens. So I was able to use that to get a pretty good distribution of the genus. As far as color goes in Pseudopolydesmus, they all are generally this sort of pink color to sort of a tan brown. The standout in the genus is really Pseudopolydesmus canadensis here in the lower left. It has this really nice black mid-dorsal stripe and almost brick red perinota along the sides. And so that's sort of the Ferrari of Pseudopolydesmus. Now, using all the specimen records that I had from my own collecting, museum holdings, and the literature records, this is the distribution of Pseudopolydesmus, sort of smoothed around the edges there. And so it occurs throughout most of Eastern North America and gets up into Canada. Now, breaking it down by species, these maps are based on about 9,300 specimens and cover 39 states and provinces. In the top corners there are the most widely distributed species, Pseudopolydesmus serratus and canadensis. Pseudopolydesmus serratus is the most widely distributed of all the species by far. And then in the middle rows here, you can see some of the other species. These middle ones are more regional in their distribution, with Pinatorum being the most widely distributed of those, whereas Erastus is more limited to the southern Appalachians and a little bit of the mid-south. And Pseudopolydesmus colonus is sort of from North Carolina up through Indiana, typically in river valleys. The 3 species on the bottom are the sort of swamp and wetland adapted species, which are much more localized in their distribution. Pseudopolydesmus minor has the widest distribution of those. It occurs from Arkansas up through Indiana. And so this is a pretty far-ranging genus, and thankfully we have a good amount of specimens held in collections. So, looking at the molecular phylogenetics, I designed 2 new primer sets for the genes 16S and enolase, and was able to sequence all described species of Pseudopolydesmus for my analysis. I used 296 specimens in these genetic analysis, including some outgroups. I used 6 different genera within the Polydesmidae, and then 6 related families within the suborder Polydesmida. And I rooted the entire phylogeny with the family Diplopoda. And so this graph on the left is a lot to take in, but I'll walk you through it. The unhighlighted taxa there are the outgroups, and then each color around the circle corresponds to one of the species. Blue is Pseudopolydesmus serratus, whereas red is canadensis, and those are the most commonly collected specimens for this project. The black branches indicate bootstrap support of over 70%, and each species within Pseudopolydesmus was well supported. So we can be pretty sure what we're seeing are the legitimate species. I've summarized it on the right with a simplified cladogram there showing the relationships of the species to each other. And to the right of this, I have a column for the morphological groups that have been hypothesized for this genus. And so the serratus group, you can see, is split between 2 different sets, whereas the canadensis group is also split. So what we're seeing is that these morphological groups are not legitimate monophyletic groups of taxa within the genus. Something that it was interesting to see was that this Raxamel phylogeny found that Serratus and Plutocollis were sister species, which is interesting because Serratus is a pretty widespread, large-bodied species, whereas Plutocollis is much smaller and is restricted to swampy areas in eastern Virginia and South Carolina. The other 2 swamp-adapted species are Minor and Caddo, which are found to be sister species. So it may be that this adaptation to these sort of wetter areas happened twice in the genus. Now, looking at the outgroups a little bit, this was something that was really exciting to me. The only previous molecular phylogenies on the Polydesmidae have solely used the gene CO1, a fragment of it, but this includes a lot more data and more outgroups as well. And so I've cut off Pseudopolydesmus here from the bottom just to make it a little bit more readable. But what this was telling us is that Apanorchotis was the most closely related genus to Pseudopolydesmus. And Apanorchotis is a genus of over 70 species of millipedes in East Asia. So that was pretty neat to see. The other eastern genus of Polydesmid in North America, Cytonotus granulatus, was found pretty far from it. So they don't seem to be particularly closely related. But these results can be considered pretty preliminary as far as relationships within the Polydesmidae go, because it doesn't include any of the Western North American genera. So it would be really interesting to see how those are all related to Pseudopolydesmus. Another interesting result here was that the most closely related family to Polydesmidae was found to be the Macrosternodesmidae. Previously, based on morphology, it was hypothesized that the Haplodesmidae were the most closely related to the Polydesmidae, but Haplodesmidae falls out quite far up here, this sort of green highlight with those 2 species of Eutrichodesmus. Also interesting is that the family Cryptodesmidae falls out in 2 separate places. One of them is the genus Q. from Japan, and then the other is Periodontodesmus, collected in Mexico. Interestingly, Periodontodesmus was previously classified within its own family, the Periodontodesmidae, and so this would be really interesting to investigate further and with more taxa. Now, looking at the star beast inferred tree using the multi-species coalescent model, we get different results. I use Polydesmus as an outgroup here, and this tree also found that Pseudopolydesmus was monophyletic, but the support values for this aren't as well supported as we saw in the Raxamel tree. And so again, this found that the species Caddo and Minor were sister to each other, and those were sister to Pinatorum, which was held by the Raxamel phylogeny as well. But down here in this other clade, it puts Canadensis and serratus as sister species, which has not been thought before from either based on morphological data or our previous genetic data with Raxamel. And so to sort of compare these in a way that you can see both the star beast and fir tree and the Raxamel, I've got Raxamel on the right and star beast on the left here. So you can see these differences in the clades that they were giving. For now, I'm putting more trust in the Raxamel phylogeny because it's more congruent with the morphology, and the fact that with our star beast analysis, the ESS values, essentially a value about how well supported the tree is, it wasn't super well supported. So I want to try running that again and seeing if I can get a better supported tree and see how that comports with the Raxamel results. Looking at the morphology, something that was done in the Searwald et al. 2019 publication is that the species Tallulanus was synonymized with Erassus. Historically, Tallulanus has been applied to a group of species in the southern Appalachians that have this large process called E2 on the gonopod. You can see it there. And so we put it under an SEM along with Pseudopolydesmus erassus. And what we found was that Erassus was previously thought not to have this E2 process, but in our SEM results, it does have one. It's just much smaller. So this is like the geographical variation, because in the molecular phylogeny, specimens of Tallulanus fall pretty well just throughout the Erassus clade. So it doesn't seem to be its own species. And so I also find support from my genetic data to synonymize these, as was done previously. I also investigated this manuscript name that Dr. Hoffman had for some species of Pseudopolydesmus in Southwest Virginia, indicated in this box down here with the dots. He called this Adocetus because the main branch of the gonopod was kind of curving like a breaching whale or dolphin. And so I went to these areas and collected specimens to compare it both morphologically and genetically. And what I found was that specimens of Adocetus fell pretty well just within the main serratus clade. And so it does not seem to be its own species, but it's apparently simply morphological variability. Now, looking at the integrative taxonomy results, this is what I got here. And so on the left, I have the morphological clusters that the modified PCA returned, and it returned 3 clusters. And then on the right, we have the taxa. And so when I first saw this, I thought, oh, well, that looks pretty good. But what was interesting was on the y-axis, we have one of the components, but on the x-axis, it goes up to 70 for some reason. And so I was looking at this and eventually realized that the x-axis is just the specimen number. And so it's just giving me the specimens as I entered them into the analysis. And so looking into it further, it turns out that the first principal component, body length, explains 96% of the variance. And so what that means is that when I made a scree plot of the eigenvalues, the first principal component explains 96% of the variance, and the next component explains 2% of the variance. And so the way that the plots were being rendered, it was just not returning the second component because it explains so little variance compared to the first. So I modified the analysis to return 2 principal components, and I got this plot. And so on the left, the clusters are, it found more clusters with 6 of them being found. And on the right, again, we have the taxa. So what we have here, and you can see that there's a very highly correlated, there's a highly positive correlation between these 2 main components. And so these morphological clusters that it found didn't really comport with the species that we're seeing, except for body size. On the left here, we have some of the smaller bodied species, and on the right, you have the larger bodied species. So these results weren't really giving us much to go off of as far as objectively delimiting species, because it was just essentially telling us which species were smaller or larger. So why might that be? Well, Pseudopolydesmus is a group that's identified mainly by the genitalia. There's not a whole lot of variation in the somatic characters, and this might be due to some of the ecological requirements. These smaller bodied species are more found in wetlands, and then these larger ones are sort of generalized deciduous forests in the east. And so with this low morphological variation, there's not a ton of information to go off of. But this method has been previously found useful in cave harvestmen and also fossil foraminifera. And so maybe in other groups of millipedes that are more variable in size and some of their body characters, particularly cave millipedes, it might work better. So to sort of talk about the significance here, this is the first multi-gene molecular phylogeny within the Polydesmidae, and it shows some interesting relationships with other families within the suborder Polydesmidia. We have 8 morphological-based species in the genus supported by the molecular data, and this data overturns the previous hypotheses about the evolutionary relationships within the genus, and that this Canadensis group and Serratus groups, they were not supported by our molecular data. And so we now have a great accumulation of morphological data and genetic data to have this stable classification of Pseudopolydesmus at last after 200 years. And so to summarize my main objectives here, I found these 8 species supported by the data. I defined the geographic distributions for each species and mapped them based on about 3,500 records. I also found that the monophyly of Pseudopolydesmus is indeed supported, and that the genetic analyses and the morphological analyses based on the gonopods are congruent. So, excuse me. So our main takeaways from each of these chapters are that afluorine millipedes have become more toxic over time, but this toxicity is affected by mimicry rings that they participate in. Appalachia has high millipede biodiversity, with 18 new species in the Nenaria wilsoni group alone, which may even constitute a new genus. And that species delimitation in millipedes should strive for multiple lines of evidence, and that molecular phylogenetics is able to solve these problems that have previously been pretty intractable between millipedes. Now with that, I want to get to my acknowledgements. First of all, I want to thank Marion Hennen, my wife, for her patience and support during this whole process. I also want to thank my committee, Paul Merrick, Bill Shear, Brent Opel, Doug Pfeiffer, and Bill Hopkins. Their comments on my projects have materially improved them and strengthened all the work that I do. I also owe a big thanks to the Merrick Lab, particularly Jackson Means, who I spent days and days and weeks in the field with, and helped me achieve a synergy with my field collection that I could not have gotten to in any other way. So thank you, Jackson, for all your help. I also want to thank the entomology department here at Virginia Tech. They've been very supportive and always helpful, and it's just great to be in an environment of such camaraderie. I also need to thank the office staff here in the department, Kathy Sheeler, Ann Jerome, and Chelsea Wang, who keep everything running and help me get whatever forms I need to turn in when I'm a little bit frazzled. I also need to thank Cal Ivanov of the VMNH and Bronwyn Williams of the North Carolina State Museum, and also Tommy McElrath of the Illinois Natural History Survey, who let me stay with him for almost a week when I was collecting millipedes in his area. So that was pretty solid of him to do. I also want to thank some collaborators, Petra Searwald, Xavier Zonley of the Field Museum, and Xavier's now at UC Davis, Joseph Ueda, who helped a lot with my questions about ancestral state reconstruction, Shahan Durkarabedian for his help on the morphological analyses, and Kurt Harden and Melissa Spring, and so many other collectors for sending me a lot of really great millipedes that help with my project. I also want to thank my funders. The National Science Foundation, through grants to Paul Merrick, funded a whole lot of my time here at Virginia Tech and my field collecting. The American Museum of Natural History gave me a Roosevelt grant that funded a lot of my field collecting, and I was able to get a grant from the Virginia Tech Graduate Student Assembly that also funded a good portion of my collecting. Also need to thank Darlene Dalton, our Latham Hall fourth floor custodian, for keeping everything clean and being a constant friendly presence in the lab. She works hard and kept everything running smoothly for us. I also need to thank all of the staff at all the national parks, national forests, state parks, and other nature preserves that I visited. I could not have done all this work without their help on permitting and questions about habitat in their parks and everything. They were huge help there. I also want to thank the Interfaces of Global Change program here at Virginia Tech, which funded my first year here, and the grad students that I've met in the program have been just outstanding. I also need to thank my Twitter science community, who has really just kept me going through a lot of this and supported me ever since I was an undergrad, really. I also need to thank the natural history community in Ohio, who really got me started not only with science, but entomology when I was doing my undergrad there, Marietta College. So they were a huge help for me. And with that, I will take any questions after I get some water, and I will check the chat and see what questions have been going on. All right, so let's see here. Got some questions. Someone asked about Robespierre maybe being a fly in the 40, Mariophora, or a relative. That could be. I'm not as familiar with forward flies and how exactly they killed the millipede. I think they're more of an internal parasite, and I think that the reports of Robespierre have been more focused on something that seems to be a vertebrate or a mammal based on like teeth markings and things. But that's a great question. Let's see. Scott Salem is asking, Chapter 1, assumption that gland size equals toxicity. Have the toxins been analyzed chemically among species? If not, okay to assume they are chemically the same? That's also a great question. So, let's see. The toxins, there's been a good deal of work about analyzing the chemicals among the species. Some work by Thomas Eisner at Cornell, I think since the 70s, has focused on some millipedes. And there are differences between millipede species, but typically within the family, the main, particularly for the Xystodesmidae, the main chemical defense is that hydrogen cyanide and benzaldehyde. And so it seems pretty likely that we can assume that chemically they're the same, at least with their main form of defense. What's interesting is that some of the smaller Polydesmida, these are in different families, not the Xystodesmidae, but for example, the Cryptodesmidae have different chemicals that seem to be more geared to deterring ant predation. And so we're lucky that Aphiloria, not Aphiloria polychroma, but another species within the genus Aphiloria, has been studied chemically, and so we can be pretty sure that this Aphiloria is using most of the same chemicals, or at least the ones that are doing the main job of toxicity. Let's see, there's another question from Matt Casson. Do you think salt shaker pore plays a role in increasing release footprint of defensive secretions? That's a great question. I would love to know more just about the chemical enzymes and everything that are involved. I'm not as knowledgeable about the exact chemical ecology of these millipedes, but I think that it could be assumed that maybe with these smaller pores, they're able to sort of release the enzymes or whatever else is being released into that reaction chamber over a wider area, and maybe different compounds than the hole type pores are releasing. Let's see, I'm trying to keep up with the questions here and not let them crawl by. Let's see, Casey Richer asks, did your segment of EF1 alpha have an intron? If so, please characterize it and how did you deal with it? So yes, the EF1 alpha segments did have an intron in both Nenaria and in Pseudopolydesmus. And so luckily, this has been dealt with in previous papers. And so I essentially followed the same thing where I partitioned the segments of EF1 alpha and partitioned the intron separately from the rest of the coding region. And that way we were able to infer it without the intron sort of throwing a lot of random data into it. Let's see. Deanie Miller asks, how consistent are these gonopod differences from these different locations? Always consistent. And so it sort of depends. With Nenaria, the gonopods can have a little bit more variation, but in general, the variation isn't very large. So typically, there might be a little bit of difference on the size of processes or the exact angle of the gonopod, perhaps. But interestingly, some of these species have variation. And so just looking at the morphology, we might even think for some of these that they could be different species. But I didn't really have enough genetic data to get to that to really assess that question in the robust way that I would like. But I think in the future, definitely there could be a project looking at just Nenaria scutularia to see if there are separate cryptic species within that, and if these morphological differences are consistent. So it kind of depends on the exact species. In Pseudopolydesmus, the variation is generally not nearly as large as in Nenaria. Like within Pseudopolydesmus serratus, for example, specimens from sort of the edges of the range, like its furthest northwest edge is in Fargo, North Dakota, and there's a little bit of difference in the size of some of the processes and the angle, but not appreciably different from much else. And so really that difference between Pseudopolydesmus erratus and Tallulanus is the largest differences in gonopods within the genus. But again, Tallulanus, based on the SEM micrographs and the genetic data, seems to just be a synonym of serratus. Let's see. Michael Palmer asks, I assume the collar is true collar rather than structural collar. I think yellow is close to impossible. Yes, so it's not structural collar. All the collar in these millipedes is based on pigment. Thomas Wiesner says, would be interesting to see how much collecting was done in the gap. I assume you're talking about the gap in the Nenaria distributions, and we have a pretty good collecting in that area. I did about a week of collecting just in Northeast Tennessee alone, looking for any Wilsonii group species. If you remember on the map, the new species Enigma goes all the way down to the Virginia border, and so I was kind of expecting that it would keep on going. I would imagine that maybe it might be found a little bit further south, but as far as I can tell, it's that gap in the distribution is a natural entity. And so it's always great to have more collecting, but right now I'm pretty sure that that is a true gap. Let's see. Victor Baranov asks, what do you think causes the disjunction of Nenaria wilsoni group distribution in Appalachia? Is it a result of glacial cycle cutting the distribution? So again, I'm assuming you're asking about that cut in northeast Tennessee and northwest North Carolina. And that's something that I want to investigate a little bit further. So near that south part of that gap, so the northern part of the southern Appalachian distributions, is Asheville and the French Broad River. And what's interesting about the French Broad River is that it has apparently cut off distributions in other taxa that are limited in similar ways as millipedes. So we've seen in plethodonid salamanders and harvestmen, Apiliones, that the French Broad creates a gap in the distributions of some species. So I wonder if possibly that had something to do with it. And again, if we can do a little bit more collecting and kind of flesh out that gap more, maybe we'll be able to get a better idea of what exactly has separated those species. Let's see. Tommy McElrath asks, if you were going to do more collecting in Georgia for cryptic millipede species, where would you go? Well, on the map, I did a lot of collecting in sort of more northeast Georgia, and so I feel pretty good about that, but I'd like to investigate a little bit more of northwestern Georgia in that area. But pretty much, and there's a good number of areas that I went to in northern Georgia and didn't end up finding them. And I think a lot of the reasoning for that is that at times when I was there, it was a little bit too dry. And so I was at Smithgall Woods State Park looking and spent about 2.5 hours there and didn't find anything, even though it was just gorgeous. It had exactly the right microhabitats I would expect. And so I bet if you were to go down in that area and just have more concerted collecting, that we'd be able to find it throughout anywhere that has pretty much deciduous forests and sort of mountainous areas. And so when it comes down to it, a lot of millipede collection is luck and just trying to be in the right place at the right time. And so if northern Georgia was a little bit more accessible, it's a pretty good drive from Blacksburg, then I'm sure we could find a ton of new species down there still. Let's see. Petra Sirawada asks, the big diversity difference between Brachyuria and Aphalloria, high diversity, and Pseudopolydesmus, comparatively low species diversity, explainable by geography of the terrain and geological history. Yeah, I would definitely agree with that. In particular, for Zistodesmidae, they have this very large radiation of species in Appalachia. Brachyuria particularly, if you recall the portion of the map that I showed, there are a ton just in the Cumberland Plateau area. And it seems to be this sort of vicariance after getting, that's been shaped by sort of the physiography and mountainous, in the mountainous type of the region. And so millipedes not being very vaginal, they can't really fly or anything, which makes it nice and easy to collect them. I think that oftentimes they can get stuck in these valleys or areas that they can't really get over a mountain or sort of really keep up gene flow, and so eventually you get these speciation events. Whereas with Pseudopolydesmus, it's got a much wider range throughout lower elevations as well as higher elevations. I think also there might be something about Pseudopolydesmus's natural history and ecology. At least Pseudopolydesmus serratus has been known to form these big aggregations and form actual migrations of millipedes. And so that might explain why it's so widespread, and maybe that's how it can keep up such gene flow between the populations to where it's got pretty low genetic diversity within the species as well as morphological diversity. Morgan Jackson asks, given your wide geographic sampling of Pseudopolydesmus, have you explored any of the population genetics and mechanisms of gene flow from Canada to Florida? Great question. So that actually gets at an interesting thing that you see with Pseudopolydesmus canadensis. And so this is typically a species that occurs higher up in mountains throughout the Appalachians and sort of into the northern US and southern Canada. And so throughout most of its range, it has it's pretty large bodied, and it has a pretty bold collar pattern of brick red paranoda and then a mid-dorsal black stripe. But in lower elevations in the the Atlantic coastal plain, you get these specimens that do not have that bold coloration. They're half the size or smaller of these mountain species, and they don't have that bold coloration. And so this has been talked about before in the publications about Pseudopolydesmus. And so it might be, it's been hypothesized before that that might even be a subspecies. And I would love to have a little bit more sampling throughout the Atlantic coastal plain and be able to investigate the different clades or the genetic differences between these sort of two morphological states that we see within Pseudopolydesmus canadensis. I've got a little bit of data on that, but there wasn't anything that sort of popped out from the genetics that was really looking like, oh, maybe this is a completely separate species or subspecies. Let's see. Miles Zhang asks, how were the genes partitioned? Odd long branch from that one species. And so the genes were partitioned in Partition Finder, and based on the different positions. So codon position 1, codon position 2, etc. It's interesting that you point out that one long species, I assume that's from the Ninnaria phylogeny, and that branch, I think, is due to some poor quality CO1 data for it. It's a species from Augusta County, Virginia. And it was a just completely new species that we've been looking for a long time. And so we wanted to get it into the phylogeny. And I think that just using the CO1 sequence that was a little bit dirtier than the other sequences sort of gave it this longer branch. And so that's what you're seeing there. MP asks, how many years have you been collecting? And for, let's see, I started collecting millipedes about 10 years ago during undergrad. But for this project, my collecting started in 2015, 2016, around that time. So this is about 4 or 5 years of collecting. And that's also added to collecting that Paul Merrick has done throughout his PhD and postdoc, and then collecting that other people, collecting of millipedes that other people have sent us. We've been lucky to have a lot of great collaborators. A former undergrad in the lab, Pat Wooden, she actually came in pretty clutch with Pseudopolydesmus. Pseudopolydesmus catto is pretty restricted to northeast Louisiana and adjacent Texas, south to the ocean, and that was pretty far to go. And so on my trip to go out there to try to collect Pseudopolydesmus catto, I wasn't able to get it. But thankfully, Pat was able to get some specimens and send them to me, so I was able to have all species of Pseudopolydesmus in the phylogeny. Casey Richard says, perhaps long branch attraction bringing Plutocollis and Serratus together in the Raxamel tree. That's a good point. I hadn't actually thought about that before. I'll need to go back and look into that. From what I recall, though, Plutocollis doesn't have particularly long branches. And I tried within the Pseudopolydesmus phylogeny to take out any that had particularly long branches, but that's definitely something to go back and check and test for. Oh, and Roger Church asked, what are the units on these trees? And those units were in base substitutions per site. Adam Blake asks, was any work done with the photographs of the aposematic color patterns of the different species at the different sites in the previous chapter? So there was a lot of work done with that. In Paul Merrick's 2009 publication on the mimicry ring, he really investigated the differences in the coloration between the millipedes and seeing how tight the mimicry was between the collar patterns. So a lot of that was previously done. Additionally, Jackson Means, as part of his PhD dissertation, did a lot of work just looking at collar variation in the in Zystodesmidae throughout the eastern United States and Canada. And so he has a great data set of what collar patterns you see on like a large level and also at a local level where the particular species are. And so there's definitely possibilities of looking closer at these sort of collar mimicry and advergence types of questions in the future. Let's see. I'm hoping I covered most of the questions there. It's kind of hard to go through the chat and do all this. All right, there's a little bit of a delay in the chat, but if anyone else has any more questions. Got a little bit of time for a couple more if you have any. So I hope you learned a lot about millipedes and enjoyed it and might want to look into them more in the future. All right, well, I think I'm going to call it there because I need to have my meeting with my committee. So thank you, everybody, for coming here and participating, and I hope you enjoyed.